Hello student, welcome to EPG Pathshala. I am Dr. Kalpana Bhargava from DRDO. So today we are going to talk about the module Basics of Molecular Therapeutics. And this is part 3, part 1 and part 2 already I have explained to you. And this is under the paper Molecular Therapeutics. So the objective of this module is to teach all of you about the advanced tool which is being utilized in molecular therapeutics. And this module is going to cover the advanced tools such as gene therapy, nanotherapy and cell-based therapy. In particular, cell-based therapy, I'll be talking about the stem cell-based therapy and all tools as well as their method as well as their applications will be discussed in the current slides. So as I said that these three therapy I'll be covering, which is gene therapy, nanotherapy and cell-based therapy. So I will start with the gene therapy. So what exactly is gene therapy? So gene therapy is a technique for correcting defective genes that are responsible for the disease development. And in fact, there are 4 approaches by which gene therapy can be utilized. The approach 1, a normal gene is inserted to compensate for the non-functional gene. This non-functional gene is actually responsible for certain disease. In the second approach, an abnormal gene which is traded for a normal gene. So that means abnormal gene is removed and it is replaced by the normal gene. In the third approach, an abnormal gene is repaired through selective reverse mutation. So the mutation which is causing the disease is repaired by a normal gene. And then the fourth approach and the last approach is the change the regulation of the gene pair. So gene pair itself is changed. So the most common approach is the abnormal gene would be swapped by the homologous recombination. And what it does, it will cause return to the normal function of the gene or the protein for a proper biological function. So it also controls the expression of the genes, which is similar to the epistasis, when one gene affects the expression of another genes. So how it all started? So this gene therapy was actually started in 1980s. Scientists began to look into the gene therapy. So what they did, they first inserted human genome genes into a bacterial cell. Then the bacteria cell would transcribe and translate the information into the protein. And then finally, they would introduce the protein into human cells. So this was an indirect approach which scientists were using in 1980s. So how this gene therapy works? A vector delivers the therapeutic gene into a patient target cell. So what is a vector? Vector is actually a carrier molecule, and usually in this case, genetic therapy is virus. So the target cell become infected with the viral vector. Then the vector genetic material is inserted into the target cells, and then functional proteins are created from the therapeutic gene, causing the cell to return to a normal state. So whatever disease cell were there, they all became normal because of the insertion of the genes. So what are the choices of vectors? So there are different kind of vectors which are being used for the gene therapy. These vectors are, some of them are viral vectors and some of them are non-viral vectors. So I will first talk about the viral vectors, or 4 types of viral vectors. These are retrovirus, adenovirus, adeno-associated virus and herpes simplex virus. These are the 4 types of virus which are being utilized in gene therapy. And then there are 2 types of non-viral vectors. These are liposomes and naked DNA, or you may also call it plasmid. So before we choose the vector system, what kind of vector system would be ideal for the gene therapy, we need to know the characteristics of the vector. So these are the characteristics which will decide is being utilized for the gene therapy. So they should have an adequate carrying capacity. They should also be undetectable by the immune system. So if the immune system detects them, then there will be immune response, and which is not good for any therapy. They also have to be non-inflammatory, so that during the insertion they should not cause any inflammation. And they should also have long duration of expression or the ability to be safely re-administered. So these are the qualities by which these vectors are chosen. So the third type of virus which is utilized in gene therapy is adeno-associated virus. So adeno-associated virus is actually a very small single-stranded DNA. So far we have seen that most of the viruses are double-stranded, but this one is single-stranded DNA, and that insert genetic material at a specific point of chromosome number 19. So it is this adeno-associated virus is actually from parvovirus family, and causes is not known disease and doesn't trigger patient. It doesn't trigger the patient immune response. So immune response is intact. So there is absolutely no problem. But there is a low information capacity. That means the information is which is carried by this virus is low in number. So the gene is always on, so the protein is always being expressed, possibly even the instances when it is not needed. And for example, this adeno-associated virus is being used for hemophilia treatment. For example, a gene-carrying vector could be injected into the muscle, prompting the muscle cells to produce factor 9 and thus prevent bleeding. So this is how hemophilia is treated, by using this vector. So the second type of virus which we are going to talk about is adenoviruses. So adenoviruses, once again, they are double-stranded DNA genome, and they cause respiratory, intestinal and eye infection in humans. The inserted DNA is not incorporated into the genome, not replicated through, and it has to be reinserted when more cells divide. So for example, common cold, these adenoviruses are derived from them. Even though all these viruses associated with gene therapy are being utilized and at different level of the experiment in animal as well as in the human model, however, there are many risks which are associated with the gene therapy. One of the risks which we all face is like they are short-lived. And what happens, they are hard rapidly integrate therapeutic DNA into genome, and their rapidly dividing nature of cells, they prevent gene therapy from long time. So basically we have to repeat it after certain amount of time. So and sometimes we'll have to repeat it multiple rounds for therapy. So that is one of the drawback. And the second drawback is immune response. So when a new thing is introduced, it leads to immune response and increases the response when a repeat offender enters. So immune response is another drawback or the challenges which is gene therapy is facing. And then the third challenge or the third drawback is the viral vectors. Patient could have toxic, immune, inflammatory response as a result of the gene therapy, and they may also cause disease once inside. So they may cause secondary disease once they are inside. Also, there is another challenge which gene therapy is facing, that is heart disease. High blood pressure, Alzheimer's, arthritis and diabetes, these are hard to treat because you need to introduce more than one genes, because these are the diseases which are actually caused by multiple factors, and that is why single gene therapy would not be enough or would not be good for treating these diseases. And sometimes they also induce a tumor if integrated in a tumor suppressor gene, because of insertional mutagenesis they cause tumor also. These are the challenges which gene therapy treatment is actually facing. So the fourth type of virus which are being utilized for gene therapy is Herpes Simplex viruses. It is actually an enveloped double-stranded naturally occurring virus that has evolved a retrograde transport mechanism and can effectively transduce sensory nerve terminal DRG neurons and the trigeminal ganglion. So as I already discussed, there are non-viral vectors also. So non-viral vectors are basically two types which are widely used. These are liposomes and DNA or plasmid. So these are the non-viral vectors which have the potential to avoid some of the critical problems which was faced by the viral vectors, and such as immune response, limited packaging capacity and random integration. So these are the challenges, these are the problems which can be avoided by using non-viral vectors. So first type of non-viral vector is liposomes. It may be mildly effective, but their activity does not last. For this approach to work, researchers need to figure out how to improve delivery, make the effect more permanent, and reduce the adverse side effect. So these are the liposomes which are at the different stages of investigation. So till date, only cationic liposome based system have been tested in clinical trials, and that too in cystic fibrosis subject. The second type of non-viral vector is naked DNA. It is also called plasmid. So what is naked DNA? It is a DNA which has a rapid clearance and has low cellular uptake. As a consequence, what happens, the main application of naked DNA injection is in vaccination and immunotherapy, and this is being used particularly for vaccination and immunotherapy treatment. So far we talked about the nanotherapeutics and how the size of the nanoparticle as well as the structure of the nanoparticle matters in treating the disease, and how nanotherapeutics are being utilized. So what will be the future prospect of the nanotherapeutics? Because nanotherapeutics are some of the medicines which are very limited and which are being utilized or which are approved by FDA. So what we want to see, what is the future prospect of the nanotherapeutics? So in near future, what we expect that oncologist and patient will benefit from suitable nanotechnology based drug delivery system that could lead to improved therapeutic outcome with reduced cost. So as the cost will reduce, it will be more and more utilized. So there are few clinical studies on oral cancer in the field of nanotechnology, where nanotechnology therapeutics is being utilized. But nanotechnology is also predicted to alter healthcare in dentistry with novel method of identifying the cancer as well as the customization of a patient therapeutic profile. So the second type of advanced molecular therapy I'm going to talk about is nanotherapy. So before we go into the nanotherapy, let me briefly explain you what is nanotechnology. So, and basically we are going to talk about the nanotechnology application here. Application of nanotechnology in area of drug delivery and therapy, it has the potential to revolutionize the treatment of many diseases. If you look at the biomedical application of nanotherapeutics, there are many, and the one which are being utilized and the one which is being investigated at different levels are, they are targeted therapy, gene delivery, drug delivery, molecular imaging, biomarker mapping, detection as well as diagnostic. So these can be used, nanotherapeutics can be used for biomarker, for biosensor, for diagnostic method. There are many of them which can be utilized. So let me just explain you what are the nanotechnology based drug delivery. So what are the benefits we have if we utilize nanotechnology based drug delivery system? So what it does, it provides multi-functionality such as targeting, delivering and reporting. It also provides improved therapeutic index. It also provides lower toxic effect and delivers multiple drugs directly to the tumor site. So there are multifunctional nanoparticle based therapies, and how they are targeted. So when we say multifunctional, that means they are targeting, they are delivering as well as they are reporting, and they are biosensing also. So everything comes in one package, and that's how this multifunctional nanotherapy based therapies are being utilized. So there are different approaches by which nanoparticle based therapies are being utilized in the current scenario, and one of the most important one is the dendrimers. So dendrimers are formed, and these are nano-sized dendrimers which are formed. So there is a medicine which is called methotrexate, and it was formerly known as the amethoprotein. Is a chemotherapy agent, and this chemotherapy agent is actually dendrite, and which is a nanomer, which are being utilized for autoimmune disease, for treating cancer and many more such diseases. The second approach which is being utilized is the nanoshell, and which is photothermal therapy. So basically molecule which is shelled by nano, it is being utilized for treating different disease. So there are uncertainties of moving multifunctional nanoparticle to the clinic. So these are the uncertainties because some of them are at investigation level in human, as well as some of them are in animal, but most of the things have not yet reached to the clinical stage, and hence there are many difficulties which scientists are facing. So first of all, define the classification. So in order to determine the characterization process ahead of the submission. So before you submit for clinical trial, you have to define the classification, then you have to provide the interfaces within the regulatory agencies, then you have to establish uniform, publicly available guidelines for the investigators, such as toxic effect and things like that. And determination of therapy is a new when it uses an existing drug on a novel delivery platform. It is a challenge. And also there is a gap which exists between the technology development and in an academic setting, and then further technology when it is taken to the maturation through clinical development and then regulatory approvals. There is a long gap which exists between these three steps. So far we talked about genetic gene therapy as well as nano-based therapy, and the last thing we are going to talk about the cell-based therapy. So what is actually cell-based therapy? So cell-based therapy is the therapy where cells are being utilized for curing a certain disease. So here we are going to talk mostly on the stem cell based therapy. So first of all, we should know what are stem cells. So stem cells are actually, these are the cells which are undifferentiated, and they may have many, like some potential scientific uses. So how the cell-based therapies are being used? They are also referred as regenerative or reparative medicine. So, how and where these cell-based therapies are being utilized? Therapeutic cloning, gene therapy, cancer research, basic research. There are many areas where stem cell-based therapies are being utilized. So, before we go into the details of the cell-based therapy, we should know what are the unique characteristics of stem cells. Why do we need to use stem cell-based therapy? So number one unique characteristics is that stem cell can regenerate, and it can form unlimited cells, and it is self-renewal through cell division. The second characteristic is stem cell can specialize. So once the stem cells are divided, and then they can specialize based on the certain function or based on the certain structure of the cells. So what happens under certain physiological or experimental condition, these cells can be specialized. They then become the cells with a special function, such as, like for example, beating cells of the heart muscle. Second example is insulin producing cells of the pancreas. So any type of cells you want it to specialize, these stem cells will specialize that particular kind of cells. So now next point we want to know is like how many types of stem cells are. So originally there are two types of stem cells, and these are embryonic stem cells, and then second one is adult stem cells. So embryonic, again, what is embryonic stem cells? Embryonic stem cells are actually received, they get from embryos which is created in vitro fertilization or from aborted embryos, and then adult stem cells. These adult stem cells can be received from limited tissues like such as bone marrow, muscle, brain. These are the tissues by which we can get adult stem cells, and they are also can be received by placental cord or baby teeth. So these are the some of the sources of the adult stem cells by which we can get the adult stem cells. Stem cells. So let us define how these stem cells are formed and how many types of stem cells are actually formed. So when you start looking at the human developmental process, there is a single cell embryo, and then it progresses to 3 days embryo, then it progresses to 5 days embryo. So the cells which we obtain from 5 to 7 days embryos are actually called embryonic stem cells, and these cells are called totipotent cells. And then it progresses to 4th week embryo, then 6th week embryo. So between 4-week embryo and 6-week embryo, if we isolate the embryonic germ cells, These are called pluripotent cells. And then there are fetal tissue stem cells which are pluripotent or multipotent. And then when the infant is there, and between the infant and adult, when we obtain the cells from different part, these are called adult stem cells, and these can be pluripotent or they can be multipotent also. So some of the organs from which we obtain the adult stem cells are cord blood stem cells or embryonic carcinoma cells. These are the cells which are called adult stem cells, and these are being utilized for different type of cell-based therapy system. So look at the different categories of stem cells. As I already mentioned, there are 4 types of stem cells: totipotent cells, pluripotent cells, multipotent cells, and unipotent cells. And all these names are derived by which these cells are isolated from the time of embryonic development, how and from where these cells are isolated. The name is based on that. So if you look at the totipotent cells, what is their characteristic? What is the definition of it? It has the capability of differentiation into all cell types. And then look at the pluripotent cells. This has the capability of differentiation into cells which are placed in fetal layers. And multipotent cells, it has the capability of differentiation into cells of specific category only. However, if you look at the unipotent cells, it has the capability of differentiation into only one type of cells. So this has a very restricted potential, and it can only differentiate into one type of cells. So as I already discussed that there are different stages of embryo by which we isolate the cells, and then they become different kind of cells like totipotent, pluripotent, and multipotent cells. So look at the difference between the embryonic and adult stem cells. As I said, there are 2 types of stem cells which are being utilized. So if you look at the embryonic cells, they are totipotent cells, and as I already discussed, that the definition of the totipotent cells is that they can differentiate into any type of cells. And then look at the adult stem cells, they can be either multipotent or pluripotent cells. So these multipotent or pluripotent cells can differentiate into some cell type. So that means there are limited outcomes. And again, these embryonic cells, their source is known, whereas in adult cells, their source is not known. In embryonic cells, the large numbers can be harvested from embryos, whereas in the adult cells, only limited number can be obtained, and it is very difficult to isolate also. The one drawback which embryonic cells has, that it may cause immune rejection, and whereas if you look at the adult stem cells, it is less likely to cause immune rejection because the patient's own cells can be used as a adult stem cells. So there are some tissue-specific adult cells also. So these adult cells are isolated from bone marrow, gastrointestinal tract. They can be isolated from tooth, especially baby tooth, and they can be isolated from skin, from hair, from central nervous system, kidney, sometimes muscles also. So these are called tissue-specific adult stem cells. So now, after knowing the different types of stem cells, now look at the therapeutic application of these stem cells in different kind of disease. So, as I already mentioned, it depends on the source of the stem cells from where these stems are isolated. Their application actually varies. So, if you look at the source, if it is from bone marrow, from peripheral blood, cord blood, these are the sources which is right now, which is currently being utilized, whereas there are some sources which will be utilized in future based on the investigation which is being carried out at different level. So these could be ESS, these could be IPSS, there could be adipose tissue also from where these cells will be isolated. And there may be some other future sources also from where these stem cells will be isolated in future. So coming to the therapeutic application, so the current which is being utilized is bone marrow transplantation, such as fractures, chronic wounds, burns, and in future we are expecting that it could be used for the myocardial infarction, heart failure, central nervous system disease, diabetes, and there are some other disease which can be treated by using stem cell therapy. So, if you look at the therapy which is being utilized, it's a regenerative therapy to treat Parkinson's disease, Alzheimer's disease, spinal cord injuries, strokes, burns, heart disease. There are many diseases which are being treated or being utilized, investigated by using cell-based therapy such as stem cell-based therapy. So, as I discussed about the homeopathic stem cells, which is isolated from the blood, as the name says that homeopathic stem cells, that means it is isolated from blood only. There are many applications of this particular stem cells, and especially in leukemias, plasma cell disorders, lymphomas, some inherited disease, and some inherited immune system disorder also. So these are the stem cells which are being utilized for various treatments. So if you look at the reality, what is being investigated and what we think is future will be. So when you look at the real scenario, there are currently only few well-established indications for the stem cell therapy, and that too, one of them is being the bone marrow transplantation. But future application will provide, hopefully, a great source of hope for many patients. And exploitation of emotionally vulnerable patient, ethical consideration in pluripotent cells. So these are the future promises which are expected actually in future. So students, let us now summarize what so far we have learned in this module. So the first point which we want to make here is that use of gene therapy is an alternative treatment, and it is going to be in a very advanced stage in the coming years. Then, given the rapid recent progress which is being observed in molecular engineering and nanotechnology field, the design of completely novel biomaterial based molecule devices and the system with function tailored for a specific application or a specific disease or a specific diagnostic seem to be much easier and more feasible than before. So now we are looking forward to much more such systems. And so far, the uses of stem cells in treatment of various blood diseases has been studied, such as lymphoblastic, leukemia, myeloid leukemia, and thalassemia. However, there are many other diseases which are being treated by stem cells, and most of them are. One of them is a popular disease which is treated is bone marrow transplantation, and some of them are being investigated at different level of research, including human as well as animal. Thank you very much.